Really appreciate the chance to present. It's been a very inspiring day so far. Really enjoyed hearing everybody else's talks. I had a sort of healthy degree of imposter syndrome when I looked at the program yesterday, and it's only been heightened by hearing everything. But I'll give it a go and share some of my work with you. So, as Esther mentioned, I'm a medical oncologist in the field of melanoma, and Ben actually introduced this quite nicely in the middle session to really speak about the progress we've made in the treatment of patients with melanoma. The advent of both sort of targeted therapies and immunotherapy has been extraordinary, and for someone like me, even in my own career, this graph on the left represents how we treated patients with melanoma with very ineffective chemotherapy when I was training in medicine. And it's kind of quite extraordinary now to be an oncologist who gets to prescribe combination immunotherapy for patients. And so, as you know, the kind of combination of CTLA-4 and anti-PD-1, this is the 5-year data here. Obviously, we've got 10-year data now. The green line, it's really quite extraordinary. And yet, I think there are some really important unanswered questions. And I think about these every time I'm in a clinic. Because the thing is, we still don't cure everyone with melanoma despite these advances. And I think the questions that I find myself being frustrated about, the ones that I can't answer for the patients in front of me, are the ones that have driven my sort of desire to kind of pursue some discovery science research. And so for me, they are really, and the themes that have come through everybody's talks today, but things related to whether we can predict for and or understand what enables metastasis, whether we can understand who might be a complete or long-term responder to some of our treatments, and then whether we can understand what drives acquired resistance to therapies and whether those processes can be reversed. And my own particular sort of scientific interests are in the area of melanoma metabolism. And so, sort of, the overarching hypothesis of my program of research is that if we take the techniques that we have to understand tumor metabolism, we might be able to apply those to really understand metabolic phenotypes and try and harness those to develop biomarkers and therapeutic combinations. And the reason I've sort of developed this interest is much like what I was reflecting on about the things that have changed in my training as a medical oncologist, is that I think that our understanding of the field of tumor metabolism has sort of changed dramatically, and particularly in the last decade or two. And so many of us, very familiar with this concept of the Warburg effect and a number of kind of observations that have been made in the field of tumor metabolism based on sort of in vitro assessment of tumor metabolism. And I think what's exciting in the modern era is that we now have the technologies that allow us to really deeply try and understand what is happening sort of within a tumor microenvironment and also within a host. And that's sort of where some of my work comes in. And so forgive me for those who sort of are aware of some of these techniques, but I appreciate that sometimes people have not thought a lot about the approaches that one might use to sort of directly assess what is happening with tumor metabolism, particularly in the in vivo setting. And so, for a bit of an analogy, a lot of people, I think, are aware of the sort of omics approaches, metabolomics or lipidomics, where you get a snapshot of the abundance of various kind of metabolites or lipid species of interest. And I sort of see this as akin to this photograph here of, counting up sort of various cars in a car park. But what it doesn't tell us is sort of how quickly a fuel might be used in a metabolic pathway. It kind of just gives us a snapshot in time. And, this other picture of this highway kind of reminds us that even when you take a snapshot, you could be fooled into thinking that a fast moving pathway is one where there's like a high amount of something, but actually that's actually the place where the traffic is stuck. And so I think thinking about techniques that allow us to truly understand how a fuel is used in tumours can provide us some additional insights. And that's where the concept of stable isotope tracing comes in. And so for those who've not seen this kind of technique, what it involves is taking a non-radioactive but heavier sort of form of a nutrient, in this case, 13 carbon labeled glucose is the work that I'll be describing today. And all 6 carbons are labeled. And so you have the opportunity then when you look with mass spec at the various metabolites of interest, very simply put here in the TCA cycle and the glycolytic pathway, to understand how much of the particular labeled version of the fuel is being used in those pathways. And so this is kind of quite a neat approach to try and understand how tumours use certain fuels. Now, the reason I've become interested in this is really that there's some clinical importance associated with this. So I had the privilege of doing some of my postdoc at UT Southwestern with Ralph de Beredinis, who sort of led this work more globally. And they've looked in lung cancer and found that patients who have a high degree of TCA cycle labeling in their tumours, that those features are associated with a significantly worse prognosis. And in sometimes it's even more prognostic than sort of more standard clinical features associated, for instance, stage. And so to me, this represents an exciting opportunity. And so in the time that I was in Dallas, started some work with a clinical sort of and translational study there alongside Ralph and Jennifer Gill, who's a physician scientist there, and a team of a multidisciplinary team, really, where we have basically started infusing patients perioperatively with this 13 carbon labeled glucose, and then taking samples, both blood and tissue, to try and evaluate them to get a better understanding of what's happening with the tumour metabolism. And so this work has been done in a few other tumour types before we started it in melanoma. And so one of the first things we did with the initial samples collected from those studies was to sort of see how melanoma compared to other tumour types. And, I mentioned earlier this sort of Warburg effect that we're all kind of familiar with, tumours being quite glycolytic, even in the presence of oxygen. And kidney cancer, clear cell renal cancer, which is depicted in the yellow here, really follows that phenotype. You can see high labeling in the glycolytic intermediates, which are on the left-hand side of this graph, and much less in the TCA cycle. But what you see in brain, lung, and now for the first time in melanoma is that there's actually quite a lot of oxidative metabolism in tumours more broadly. In melanoma in particular, we seem to see a range, even in a very small number of patients, of some that have low and some that have high labeling. And so it's really early days with that data. But even looking on the first 8 patients, when we look at this measure of the labeling in citrate relative to pyruvate, so we see this kind of as a surrogate of glucose oxidation. You can see in the blue here, those with a higher degree of this ratio, that seems to be with associated with an increased, relapse rate within 12 months. And so it's very early data yet, but it does seem like the findings in lung cancer might be seen here in melanoma as well. One of the other really intriguing findings that we've had in that early data set is that when you look at the amount of labeled glucose in the tumour relative to the plasma, you find that when that ratio is small, or lower, that seems to also be associated with a higher rate of relapse within the first 12 months. And, this is something we're really thinking about in terms of whether it reflects low uptake of glucose, whether it means that there are alternative fuels for the TCA cycle, whether it represents tumours in which there's gluconeogenesis and therefore more unlabeled glucose. And so this is something that we're really interested in as well. Which brings me then to this project, which is really a proposal to apply these techniques in the setting of treatment. And so we've talked a lot about predictors of response and resistance, I think, to therapy. And that's very much the approach here, which is to try and look in a initially small, but over time larger group of patients, either prior to having adjuvant therapy for melanoma or in the context of resistance to therapy. And in this cancer grant, I had also proposed sort of a bit more of a transcriptomic analysis to go alongside it, because I think, as Aretha illustrated before, much more in a much more sophisticated way, many people make inferences about the metabolism of a tumor or immune cells based on gene expression or proteomic data. And I think what's missing in the field sometimes is a direct assessment of metabolism, and coupled with some of the more conventional ways that we might try and assess metabolism. And so we're hoping that by kind of doing that in a more direct way, we'll start to get some insights into, how the 13C data might link with transcriptomic analysis. And so what has been a pretty challenging endeavour over the last few years is for me to establish a program of stable isotope tracing here at Peter Mac for many reasons. But I think particularly just because it was something new. And so it's been a real multidisciplinary effort, and I can't go through all of the people involved. But you can see that this is a range of people, particularly surgeons, anaesthetists, pathologists, and imaging specialists. So we're really thrilled to have finally gotten this off the ground here. And so what I can share from you, which is data generated as part of this Cure Cancer project, is really from the first 8 patients that we've done infusions in. And it's too early to be able to associate this with, relapse data or prognostic information. But I think given what we knew in that sort of therapy naive setting from Dallas, it's intriguing to know that we can kind of, map out here in this graph patients that sort of have low to high labeling in the TCA cycle, and start to correlate that with things like their oncogenic mutation status, the underlying driver, the site of disease, and also, more importantly, those who've progressed on treatment or not. And so for what it's worth, this sort of very high labeling in citrate relative to pyruvate has occurred in a patient who has progressed on both targeted therapy and immunotherapy. And so it'll be interesting as we get larger numbers to see what that means. And I kind of have also taken the time to have a look at that feature that I mentioned just earlier about the glucose uptake relative to the plasma uptake. And again, sort of we see another range, and do see some of the patients who've had multiple lines of therapy sitting at the more left-hand side of this graph, so a lower kind of amount of tumour, labelled glucose relative to plasma. And so this is something of interest for us. This is extremely early data, but we've just sort of had our first run of the single nuclear RNA-seq that we're doing in parallel from these samples. And I'm no bioinformatician, but I'm working with a bioinformatician in our lab, and just using a very automated approach. These are some initial UMAPs, but obviously we're going to spend a lot of time with the clustering and annotation here. But I think that there will be a lot to be gained by sort of more deeply looking at the immune cell composition, but more importantly, the metabolic gene expression in different cell types, in different contexts, therapy naive and resistance, and correlating that with the stable isotope tracing features. And so that is work to come in the coming months, and we're pretty excited about that. So I guess I will just sort of wrap up by reflecting on the fact that for a long time we have thought about the Warburg effect in cancer, and whilst it's often important in cell culture models, I think the approach of looking really directly in a tumour with stable isotope tracing is allowing us to understand that the TCA cycle is quite important and possibly very prognostic, maybe predictive of therapy response, and particularly in melanoma, but some other cancers. I think this is proving to be an important area of cancer biology that I think I'm excited to kind of deeply dive into over the next few years. So with that, I just want to acknowledge that, I started this work in the lab of Ralph de Berardinis at UT Southwestern and have been continuing it under the mentorship of Grant MacArthur here at Peter Mac. In both cases, it involves a lot of input from many collaborators, so both study teams across both sites. But more than that, this is a very altruistic type of cancer research. A patient is agreeing to have an intervention, an infusion of something they don't need, and multiple samples. And I think it's kind of quite extraordinary how many patients are willing and excited to participate in this kind of study. And of course, to acknowledge the funders, particularly Cure Cancer. This funding came at an important time for me when I really needed to get this work off the ground, and it's allowed me to kind of look a little bit beyond metabolism with the transcriptomic work. So I'm pretty excited about that. Happy to take any questions.